Hey guys, this is the answer key for the mutation activity that you're going to do earlier. And this is very similar to the worksheet that you're going to have to do with your weekly activity or your weekly project. So let's go over this and let's go through how to do it. You're going to do it very similar to last week's when we did the alien lab where you'd have your links. And so remember that with mRNA, instead of T's, you're going to have U's. So when we look at this, when we translate it, we're going to actually have an a U, a G, a U, a U, an a U, a C, a U. C pairs with G, and then the G's pair with C's, T pairs with A's pairs with U's, and C with G and T with A. So that is your code for that particular mRNA strand. Then using your codon sheet, which you have at the bottom of your page, you're going to find these different codes and you're going to write out your protein linkage for the amino acids. So remember you have to have an AUG to start. So here's your first AUG right here. Okay. So you're going to use your chart and you're going to find AUG, and it's MET is what your amino acid linkage is to it. Then you look at your next codon, which is, remember, is every 3 letters. So you have UUA. So you're going to go up here to U, down to U, and then over to A, and it's leucine, LEU. And then you have your next 3 letters, which is UCU. So UCU with this serine. And then you have your GCC. You have GCC, and that is alanine. And then you have your AUU. So AUU, just allene. And then you have your last codon, which is right here. So that's U over to G, A, and it says stop. So that's where your protein would stop. So this would be what your amino linkage should look like, and this would be what your final protein is. Okay. So now what we're going to do is we're going to start looking and changing some things from the original DNA strand to see what happens. So this says change the 7th nucleotide in the original DNA strand from a T to a C. So we count over 1, 2, 3, 4, 5, 6, 7. So this is no longer going to be a T, it's going to be a C. And then the rest of the letters we're going to write down the same. We're just rewriting that with the mutated form. Not changing anything yet. Oops, did that automatically, sorry. Okay, so then you're going to write your mRNA strand. So T's are A's, A's are U's, C's are G's, A's are U's, C's are G's, and then you have your A is a U, G is a C, A is a U, C is a G, your G's are C's, your T's are A's, your A's are U's, C is a G, and T is an A. So that is what your mRNA strand will look like. So now you're going to do your mRNA sequence. So you're going to use your chart when you do this and find your AUG. So AUG is MET because that's your start amino acid. And then you're going to have your UUG. So UUG is leucine. And then you have your UCU. And UCU is serine. And then you have your GCC, and that's alanine. And then you have your AUU, which is allene. And then you have your UGA, which is a stop sign. So from this and this, the amino acids are the exact same. So this didn't cause any mutation that would cause harm or cause a change in your DNA strand or your protein strand. And so this kind of mutation is called a silent mutation because it did change, it did mutate because that T changed to a C, but it didn't have any effect on the protein because they all paired for the same amino acids. So that's called a silent mutation. Okay, so the next one says that you're going to add a G to the original DNA strand after the fourth nucleotide. So here you have T, T, A, C, and then you're going to add a G inside of there. So you're not going to get rid of anything, you're just going to add something. And remember, you're not changing any of the letters at this point, which is hard to do when you're thinking about it. So what's going to happen is you're going to have this codon, or this code for your mRNA. And you're going to have an extra letter here at the end. Okay. So remember that we have to start with our AUG. So we find AUG. We know that that's always going to be the same amino acid. We're going to look at CUU as our next one. And that's using the chart we find out is leucine. Sorry. And then AUC is ileum. And then UGC, cysteine. CAU is histidine. And then UUG is LEU. And you don't have a stop codon, you just ran out of letters in this particular sequence. Okay? So because you didn't have that final codon and you added that letter, everything sort of shifted. Okay? So you had some of these different proteins, amino acids would make the protein different and maybe have a different function. This kind of mutation is called a frame shift mutation. Now remember, I'm getting my amino acids from looking at this chart. So every 3 letters you go and you look at the chart and you see what you have. Okay. All right. So the back page. This one says change the 6th nucleotide in the original DNA strand from an A to a T. So we count over 6. So that's going to change right here. Is going to change to a T. Okay. So I'm going to write that down. So it's going to be exactly the same. You're not changing anything yet. And then you're going to do your mRNA strand. Okay, so you're going to have your mRNA. Hopefully you're getting really good at doing this. Oops, that's a U, sorry. Okay, so you find your start. So that is your amino acid always. And then you've got UAA as your second one. So when we go to the chart and we find UUA, we get, it's UAA. Right here, we get a stop. So our protein would stop after that first amino acid. Okay, so that wouldn't make any sense. So the type of mutation is called a nonsense. Okay, that's how I remember it is nonsense. Doesn't make sense. All right, so on the next one, you change the 12th nucleotide from a G to a C. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So you're changing that G to a C. So again, you write down the code without any changes at this point. Okay, and then you're going to change it to your mRNA. So remember, you have A's. And so you have these different letters here. You look for your start codon, and then you follow every three and finding out your amino acids linkage. Until you get to a stop. And it does change the amino acid linkage, and so therefore it does change your protein by adding that, changing that letter. So this is called a missense mutation. Okay. So the last part starts dealing with different blood cells, and this one deals with your red blood cells. It says red blood cells have a protein called hemoglobin that carries oxygen. A mutation, the hemoglobin gene, causes the disease sickle cell anemia. Below is the portion of the hemoglobin gene where the mutation is found. For each strand of DNA, write the mRNA sequence, circle the mRNA codons, and write the amino acid sequence. So we basically are looking at our normal one. We're writing our strand out, which looks like this. And I wrote these a little large, so we're sort of going to the end of the line here. And then I use my codon chart to write out the amino acids. And that's what you should have gotten. And then the last thing, it wanted me to do the codons, so I'm going to put boxes around the codons. Every three letters is a codon. And the last codon should signal stop, but in this case it did not. All right, so the mutated hemoglobin DNA is resulting in the sickle cell anemia is this one. So we're going to compare these two after we get our codons wrote out. And then we do our chart and we get our amino acids. So we have MET, which is our start. We're going to put squares around our codons. And we notice that we have everything alike until we get to the end. And this one changes to valine, which is from the GLU to VAL. And then the last one is the same. Okay, so there is a difference. So one protein has been coded wrong, and then the T has been changed to an A, is what the difference is between the normal and the sickle cell anemia. So here we write that the protein has been coded wrong, and the T has been changed to an A. And then the last question says, what specific kind of mutation causes sickle cell anemia? And it's the missense mutation. Okay. So these are the answers to this worksheet. Make sure you go back and you look over the worksheet and make sure you can do all the different steps. and this will help you with your weekly project. I hope this helped you figure out what you needed to do, and I hope to see you guys soon.